Hi, let's continue on with the second part of molecular genetics. So as we saw earlier on, we saw how you have got the phosphate backbone group of DNA itself, and we also saw how the nitrogenous bases, they can form hydrogen bonds. So even here, you can see that the nitrogenous bases, they are forming hydrogen bonds together. This is the part whereby they are winded around to each other already. And at the back of it, these sections here, these are the backbones consisting of my pentose sugar covalent structure and also my phosphate group's covalent structure. So what you can see is that earlier on when we talk about how they are in opposite direction, so meaning to say that they are running in anti-parallel. So if one strand, this one is the 5' end, if you were to continue it down, it will end up over here to be the 3' end. So in other words, the opposite structure over here, opposite strand will be 5' end. Over here at the top will be a 3' end in anti-parallel direction. So they coil around each other in this direction, and we were to zoom in on one of the section of it, we can see that the phosphate group over here and the sugar group, they are covalently bonded and forms the backbone of it. While the nitrogenous bases, they can form hydrogen bonds in between them and as they intertwine around each other. So when you do DNA replication, this intertwining section itself, they're going to unwind just a little portion that is supposed to undergo replication. Let's say, for example, if you have a tear in the skin or the muscles and your body have to repair, and the DNA will send out the information for the replication. So for that, we will look at it in the Central Dogma of DNA. So before we go into that, let's look at what is determining a gene, or what is exactly a gene. Okay, so in the topic for cells under organelles, we know that we have got cells, the nucleus in almost every cell of our body system. So not all cells do contain nucleus, but majority of it do contain it. So within the cell itself, we have got nucleus. Within the nucleus, we have got the nucleolus over here. Now the nucleolus is the important section because within the nucleolus, we have what we call the genes within it. But what is a gene? So every individual one of us contains what is known as chromosomes. So in humans, we have got 23 pairs of chromosomes in each and every one of us. So within these 23 pairs of chromosomes are millions of data that give rise to who we are and what we are. Okay, at least in an anatomical sense or point of view. So these 23 pairs, every one of these pairs will have different segments within it. Okay, so let's say, for example, this particular segment that you see in blue here, there will be other segments that are running through it on the entire segment of the chromosome. Each of these segments will code for a specific information on you or in you in terms of maybe your organs or how your system is running, the biochemical pathways that your body is taking. Okay, so let's say take for example over here, the gene. Okay, so this part of the gene is a specific location, and this specific location is what we call a locus. So on this particular chromosome, on a specific region, a specific locus on that chromosome contains a series of DNA information. And this segment of the DNA information is what we call a gene. Okay, so we have got millions of base pairs of DNA within us. Definitely not all the millions of base pairs are going to code for only one thing, otherwise it will just be a single cell organism. So meaning to say, whatever that we have now, whatever that we are experiencing is actually coded by all these millions of base pairs, and different segments are going to code for different things. So if one segment of the gene over here is comprising of maybe a few hundred base pairs, it will code, for example, the brown color. Okay, so likewise, another segment, another location or locus is going to code for the hair color. I might even have another location or another locus over here that is coding for the texture of your hair. Do you have straight hair, curly hair? How dense is your hair or how fine is your hair? It is all coded within the genetic information. Okay, so how is it possible that these little base pairs actually give rise to what we have, our organs, our hormones, the proteins that we produce? And that is covered under what is known as the Central Dogma of DNA. So now, Let's recap back or review back over here. So we have got our nucleus. So within the nucleus, I'm just going to draw the nucleolus really big. So within the nucleus, I've got my nucleolus itself. So in the nucleolus contains all my genetic information, which is called the DNA. Okay. Now the purpose of the nucleus, if you recall, is to control cell activities. So basically the nucleus or the nucleolus is the brain of the cell. So the nucleolus will send out a specific information that tells, so let's say for example, you scratched yourself or you have got a muscle tear and that area requires repairing. So what it does is that the brain of the cell, the nucleus, the DNA sends out the information for repair. But of course, by itself, the DNA structure and let's say a protein structure is two completely different structures. So how does the nucleus do it? That is the question. So in order for the nucleus itself to bring the information for repairing instructions, it have to send out a message. And that message is sent out by the messenger RNA. So M stands for messenger. So this messenger RNA itself. So over here, I have got my DNA. Okay, so once I have got my DNA strand, let's say for example, I have got this section which is coding for the muscle that I want to repair. So this particular section of the gene, which is located on a specific locus of the chromosome. So let's say this is the locus. This is my gene itself, this whole strand of the base pairs. If I were to read it out, I will have this entire series of the base pairs here. So this section in red, it is just the phosphate group and the pentose sugar which forms the backbone. Okay, so over here I have got the bases here. So these bases will undergo what we call transcription. Okay, so in order for the message to go out from the DNA, it has to be somewhat different because the message that is received by the cytoplasm, it speaks a different language from the nucleus. Okay, so in order for the message from the nucleus to enter into the cytoplasm, I have to tweak it a little bit. So some of the components are still similar in the sense that they are just following the base pairing rules. Okay, so if we recall, A will always base pair with T and vice versa. C will always base pair with G and vice versa over here. So T will base pair with A over here. Now A by right, it should base pair to T, but because we are looking at RNA, remember in the earlier part of the video when we talk about how the T is being replaced with a U over here. So instead of T in RNA, the T is replaced with a U. So in the same way over here, okay, so A by right it should base pair with T, but instead it becomes a U simply because it is a mRNA. Okay, everything else, the rules will still follow through. C will still base pair with G, and I have got a G over here, it will still base pair with C. So the only difference really is A, instead of T, it becomes U. Once the DNA transforms the DNA information, or rather transcribe, because it is the process is called transcription. So once the message from the DNA is transcribed into mRNA, the mRNA will then leave the nucleus to enter into the cytoplasm itself. Okay, so once it enters into the cytoplasm, all these languages have to undergo translation. It has to be translated into the type of the amino acids and subsequently the type of proteins that we want in order for the repairing to take place or for the synthesis of a specific protein to take place. Okay, so over here, each of these components, every 3 sets of it, okay, is actually one specific amino acid. So if you recall back your topic, proteins, polypeptides to amino acids. So in the same way, every one of these amino acids, they are actually coded by 3 of these base pairs. Now we don't have to go and memorize every of these 3 components. It will be given to you in a table. You just have to learn how to identify it. Neither do you have to go and memorize the structures of these 20 different amino acids. Okay, what you do have to know is that 3 of these base pairs on the mRNA is what we call the triplet codes. Triplet because there are 3 of it and it codes for one specific amino acids by itself. Okay, so where are all these amino acids synthesized? That will require you to recap back on the topic on your cells itself. So once the mRNA goes into the cytoplasm, remember in the cytoplasm we have got the rough endoplasmic reticulum, the RER. So the RER contains ribosomes over here, which is why the translation happens on the ribosomes. So the purpose of the ribosome, okay, is to synthesize these set of the polypeptides over here. Every amino acid will then come together to give you a specific polypeptide. So once I've got the polypeptide, it becomes a protein, and it will then be sent to the Golgi apparatus or the Golgi body. So at the Golgi apparatus, protein modification takes place. Protein packaging into secretory vesicles will also take place. So once it is packaged, the secretory vesicles will then bud off from the Golgi apparatus, bind together with the phospholipid bilayer, and the proteins will be released out of the cell via exocytosis. Now, the same ribosomes will also be used if it is for intracellular use, just that free ribosomes will be utilized over here. Okay, so let's take a further look over here. So we know that the DNA itself, we have got two strands. So I have got a 5' strand for the DNA, and I have got a 3' strand, which is the top strand over here. So because they are anti-parallel, I have got my 3' strand on the other side, and I've got the 5' strand on the other end. Okay, so when by right, these two DNA all will be intertwined. Okay, but during the process of DNA transcription and translation, which is what you saw over here, the section of the DNA that is supposed to be used, this segment of the gene, will unwind itself. Okay, so once it unwinds itself, now I have got the top strand, which is 5' to 3', and the bottom strand, which is called the 3' to 5' over here. So the 5' to 3' strand over here, this is the parent strand, but this bottom strand is the one that is being used for transcription, which is what you see here, the 3' to 5', okay, which coincides with this over here. And this strand that is used for transcription to form the mRNA is what we call the template strand. Okay, so what you will be expected to know is that if this one, let's say this one, this is TACC. So let's say this one, I have got TACC over here, exactly the same as what you see in the notes. Then the parent strand over here at the top, the 5' to 3' of the DNA, it will be exactly just the base pairing. So A base pair with T, then A base pair with T over here, C with G, and the other one is also G. Okay, so from here you can derive the mRNA, because the mRNA will be using the template strand, which is the 3' to 5', and that is what you get over here, which we looked at just now. Okay, so since each of these three specific triplet codons, they code for one specific amino acid. So as long as you have got the change in even one of the base pair, so let's say for example this particular one, let me shade it in blue. Okay, so let's say this particular A is changed to maybe a G, then I will have a change in my amino acid, and that is what we call over here, once a mutation happens in the triplet codon, the entire amino acid structure will be changed. And because amino acids, each of the amino acids, they have got different types and forms into how they can form into a three-dimensional shape. By changing one of the structure of just one amino acid, it can affect the entire protein structure, which will in turn affect the entire function of the body. Okay, so the next part that we will look at will be called the genetic engineering into how we can actually produce human insulin. Thank you for listening.